Thank you very much. Thank you, everybody, for coming. So I'm going to be talking about a sensor today. So quick question first. Does anybody know about a sensor that you use every day, but you're probably not aware of it? Yep. Oh, OK. I know what he's talking about. This is the motion sensor. So in the lights, when they shut the lights off after you're done. Yes, absolutely. So that's another sensor. One other one that I was thinking of is a smoke detector. But we use sensors a lot to tell us about our environment. Sometimes, as was mentioned there, that we use it to then actuate a machine. But in a lot of cases, what I'm going to be talking about today is a sensor that has applications for safety. So a lot like the smoke detector, telling you whether or not there's a hazardous situation. So the nano part of it is that some of the applications for this are in testing the safety of nanomaterials. Now, something about this sensor is very different from all the other sensors out there, which is it uses a level of intelligence that we all possess within ourselves, and that is cellular intelligence. And I'll get into this later, but the key to this sensor is it can read out how a cell is reacting to its environment. So we use living cells, which is not normal. Most sensors are going to use an inert object. Now, how do you go about using cells in a sensor? Well, cells will react to their environment, and they can do a number of different things on this. One thing that they can do is they can change their shape. It's very common that a cell, when it's under stress, is going to form into a round ball-like shape. When they're happy and they're unstressed and they're just kind of chilling out there, they flatten themselves out onto a surface like a pancake. Now, the other thing that cells can do is, a lot like us, if they don't like where they're at, they move. So they can literally crawl across the surface and get away from something. And of course, the last thing that can happen to a cell is they can die. So this sensor is designed to sense all of these different behaviors in the cell. Now, there's a key thing to understand about how we detect this. We detect mechanical changes in the cell, how stiff it is, and other aspects such as that. And cells are very interesting. If you've ever lived in a house that had remodeling, or you had a house built for you, you surely saw the structure within the house, all of these studs and pieces of wood. That defines the rigid structure of the house. Now, cells actually have very similar structures to this, because if they didn't, we'd all just be blobs of water, and we wouldn't even be able to move across a floor, never mind anything else. So the picture that you see up here on the right is a fluorescence microscope image. So what happened is all the structures within the cells were painted with special dyes that will attach only to particular structures in the cells and light up in different colors. So what you see on here is these little blue circles in the center. That's the nucleus. It contains all the DNA. And then on top of that, you see these two other green and yellow colors. These are actin filaments, which are a lot like the studs within the house. And the other ones are microtubules. Microtubules, for anyone who doesn't know, are just basically the highway system within the cell. This is the transportation network that the cell moves materials in and out through it. The interesting part about it, though, is that unlike a house where all of those structures are fixed, the cell can dynamically tear these down and rebuild them. So within a matter of minutes, it can change its shape and change how stiff it is in response to the environment, trying to get respond to something. Now, why do we particularly use cells? Well, cells, I said before, it's an intelligent sensing system. The key to this is that they know if something is toxic. Now, how many people have been stung by a bee before? Probably just about everybody in the audience. Now, unfortunately, of course, some people are allergic to bee stings, but the rest of us, what happens when you get it? You get a welt. Now, that's not because your brain is sitting here saying, oh, I got stung by a bee, I better swell. This is because the local cells are responding to the toxins released in the bee venom, and they are changing their shape. You can physically see that because you get a swelling in there. So this is an indication that we can use cells as something that they know what their environment is. They know whether they're happy or not. And we have ways then of being able to tell. In one application, materials, toxicity is generally a bad thing. If I came over and said, oh, I've got a bucket of plutonium over here, most people would probably get out of the building. or asbestos, or other things like that, toxic waste. These are all things that we intrinsically understand are not the kind of stuff we want to be around. So generally speaking, when you're making a new material, you need to know whether it's toxic or not. And in particular, nanomaterials, we make so many new nanomaterials that we just don't have the ability to test all of these for their toxicity. But we've learned our lesson from previous times. We know we have to find out if they're toxic before we use them. So therefore, we need methods that allow us to rapidly and accurately tell us whether they're toxic. And that's actually one of the applications for this sensor. Now, another instance where toxicity is in fact a good thing is in medicine. Now, you might think, well, hang on a second. Last time I took an aspirin, I didn't want to get sick from that. That's not what I'm talking about, but there are cases where one does want this. If you have a circumstance where chemotherapeutics, for example, the whole purpose of a chemotherapeutic is to kill off the cancer cells. So that's targeted, but you need to know whether it's going to work or not. Now, unfortunately, as it is right now, doctors don't really have a good idea of whether a particular drug will work for a particular person. It's regrettably something that one just has to guess. That's not good because, first off, those drugs are very expensive. Second of all, you're dealing with someone's life where they are on a timeline. You don't want to have to wait 3 months and come back and say, sorry, this one didn't work, let's try another one. So the other thing that this sensor can be used for is to take cells from this person and test whether their cells will react to that particular drug. It's called customized therapeutics. And it's a very important revolution in those kinds of cancer therapies. Now, we have to be able to find a way for the sensor to talk to the cells. And the question is, how do we go about doing that? The key thing to this sensor is it finds out two pieces of information. The first thing is it finds out how heavy it is. Now, it's not really that the cell changes its mass, but rather it changes its distribution. If you remember, I mentioned it goes from a ball shape to a pancake shape. The way it sits on the sensor influences how much of its mass will influence that. It's a lot like when you go out walking in snow. If you wear snowshoes versus regular shoes, it determines whether or not how heavy you appear to the snow. The other thing we can do is find out the stiffness of the cell. And those two pieces of information will give us a lot to work on to find out how healthy the cells are. Now, the nature of this particular sensor is we use something that all of you have either on your possession right now or have used on a daily basis. And it is something called a quartz crystal. Now, if you've got a watch, unless it's a wind-up watch, then you have a little piece of quartz inside there shaped like a tuning fork. So the one on the left there. That vibrates back and forth using a method called piezoelectricity. The idea is that when you squeeze the material, it produces a voltage. If you apply a voltage, it changes its shape. And so a material that will resonate, so think of like a wine glass that you plink and you hear the tone from it. Same thing here. This will vibrate at a very specific frequency and it can produce an electrical signal that can then be read out. Now, the one that we use is not the tuning fork because it's hard to get cells on that, but we use the one in the middle, which is a plate-like structure, but it works the same way. It moves back and forth like this and it generates a voltage. You, in fact, all have these in your possession, probably not here though. It's inside your computer. This is what determines the frequency that your computer operates at. Now, what we do is we put the cells on top of this, and this thing is moving back and forth like this, so it's shaking the cell. How heavy the cell is weighing on the crystal, and how effectively the crystal is transferring its energy to that cell tells us the information that we need to know. Now, an important thing to pay attention to is something called resonant frequency. And everybody who has ever sat on a swing is familiar with resonant frequency. If you swing your legs at just the right speed, you will gain amplitude in your swing. If you swing too slow or too fast, you won't go anywhere. So it's important then that you be able to get to this resonance. And the quartz crystal is specifically prepared so it has a very specific frequency that it resonates at. In our case, it's about 10 million times per second that it oscillates back and forth. Now, once we put cells on there, the frequency changes. We can also detect how hard it is to move the crystal because of those cells being on there. we want to find out some information about the structures of the cell, and stiffness tells us a lot about this. Now at this point I'd like to get 2 volunteers that I've actually arranged in advance to come on up to the stage, because what we're going to do is we're going to try a little experiment here. So come on up here. Now I'm going to give each of you a Jell-O cup that's going to represent a cell. My favorite flavor of cell, by the way, raspberry. One of them is going to be stiff because it has the plastic casing on it. The other one is going to be all wiggly. Okay, so you two come on over here. All right, now go ahead and face the front here, and I'm going to hand these off to you, and then I'm going to give you some instructions on how to do this. I'll give you yours in just a second here. All right? Now the idea is that these are not all that different in their mass. The cup doesn't really add much to this. It's all about the stiffness. This one, with the plastic on, is nice and firm. So a very simple thing for both of you to do. Why don't I come up here just a little bit more. You can move back just a little bit. Now I want you to just gently, because we don't want Jell-O flying all over the place, gently jiggle these back and forth, and I want you to tell me whether they feel the same or different. So is it harder or easier to move?
Not easier.
Okay. And how about you? You think that's... Are they equally easy, or are they different on how easy it is to shake? Okay, well, we have a bit of a disagreement on whether it's easier or harder, but the key thing to this is that you see there's a difference in this. Okay, well, thank you very much. Why don't we thank our participants here? All right. So, same exact thing is happening with this quartz crystal microbalance, which is the term that we use for the quartz crystal. And in that, then, we can detect how much energy it takes to be able to shake the cells back and forth. And we can read that out. Now, I'm going to give you an example of a typical output that we get from this. What you're seeing here is a change in frequency. So the crystal is nominally oscillating at 10 megahertz, so 10 million times per second, with cells then added on it at the first arrow there. And what you see is the cells, when they land on the crystal, start weighing it down. So it's a lot like if you get on a swing and you're a bigger person versus a small person, the bigger person is going to go back and forth more slowly because there's more mass. So we see a decrease in the frequency at the rate at which it's oscillating back and forth. Now you notice that it comes back up, and the reason it comes back up is because the cells have landed on the surface and they're now making themselves at home, and they're flattening out, so the frequency goes up a little bit more. So there's where cells are added, the cells are rounded, landed on the surface at this point, and now they start to flatten themselves out over here. Now I want to give you an example related to nanotechnology here. Carbon nanotubes. These are a very interesting, neat material, but there are questions about whether or not they're toxic. And so what we have done is we have taken cells that exist inside your lungs. These are called macrophages. If you're not familiar with these, they are something that makes your life much easier. They are the garbage collector of the body. What they do is they crawl around and they gobble up all of the garbage that's sitting in your lungs. Think about this for a second. When was the last time you looked under your bed? Now, unless you're one of those people that cleans every week, you probably found dust bunnies under there. Now, did it ever occur to you that you're breathing the same air that creates those dust bunnies underneath the bed? So why is it that our lungs are not chock full of lint within a few months of us being born? The reason is because these macrophages run around, gobble up all of this stuff, and digest it for us. Now, the problem, though, is that they don't really know enough about what they're eating. So they're kind of like that friend of yours that will eat anything you put in front of them. So if you put carbon nanotubes in front of them, they'll eat them too. Problem, though, is that the carbon nanotubes are not dissolvable by the methods that they have, and they're long and pointy. And macrophages look like water balloons. When was the last time you brought a pointy object near a water balloon? It pops. And that's a bad news, because these cells have all of these enzymes which are intended for digesting materials that then spread over your lungs. So much like asbestos, which produces something known as mesothelioma, you get scarring on the tissue of your lungs when you get exposed. So we tested the carbon nanotubes to see, are they toxic, and at what concentration are they toxic? The answer, unfortunately, is that yes, they are toxic, but we know how much the exposure limits are. So you understand what you're looking at. You're seeing, again, a fluorescent image of the cells, but the green that you're looking at are carbon nanotubes, and the top row is something that the cells actually like to eat, and it's safe for them to eat. This is basically their cell food. So you can see this zymosin A is something that's okay for them to eat, and after 24 hours, they've digested the majority of them. Now, SWCNT just means single wall carbon nanotube. And we have two different concentrations, a large concentration at the bottom and a medium concentration in the middle. You'll notice that the cells already gobbled up all of the carbon nanotubes. But over time, you'll notice that at the 100, we have less cells. Where did they go? Well, they popped. Now, just to show you the kinds of responses we get, if you look at the top left side, this is just the cells sitting there enjoying themselves with nothing there. Now we start giving increasing dosages of the nanotubes. And what I want you to pay attention to is when the frequency, which is that black line, goes down, that means the cells are responding. If they don't come back up, then that means that they're not going to make it. And you'll notice at 3 micrograms, they react, but then they go back up. At 30 micrograms, they go down, but they come back up. 100 micrograms, they're down for the count. So that tells us that 100 micrograms is not a safe limit, but 30 is okay. So we know some exposure levels, so people working with carbon nanotubes know in advance what the risks are and how much exposure they should tolerate. So that's everything I wanted to say. I just wanted to put up a slide of the colleagues of mine that work with me on this. So Abish DeWild, Jianping Zhang, Gavin Wang, and Leon Dai, these are the students that work on it, and my colleagues in biology, Susan Braunhut, and chemistry, Ken Marks, without whose help we wouldn't be able to do this. And I thank you very much for your attention.
Let's give a warm round of applause. Thank you very much.